All right, human A and P, we're about to go over some vision. All right, so this is everything you need to know for vision for the light pathway. And we're gonna go over the dark pathway next. But this is everything that happens during the light pathway. All right, so first thing first. All of this is gonna go on in a photoreceptor, which is located in the back of the retina. You have light, which brings in energy as photons, is going to come in and is going to hit the visual pigment in that photoreceptor. You have what's called rhodopsin, okay? Rhodopsin just means retinol and opsin combined together. Retinol is the light absorbing molecule, opsin is the membrane protein. And in order to disrupt this and change the configuration of it, you need energy, you need photons. And this is exactly what the light brings. So when the light hits this, it triggers a shape change in the retinal from 11 cis retinal to all trans retinal, releasing the retinal from the opsin. All right, so that's step one. Step two is once 11 cis retinal transforms into all trans retinal, once that transformation happens, it activates the visual pigment. And the activated visual pigment activates transducin, which is a G protein, okay? Step three is the activated transducin is going to activate the phosphodiesterase, abbreviated as PDE, okay? Then steps 4 and 5 is when the activated PDE is going to pull cGMP off of these guys, which are called cGMP gated channels. So it's going to pull this off of that. And when that happens, it causes the cGMP gated channels to close shut, preventing positive sodium ions and positive calcium ions from coming into the photoreceptor and depolarizing it. This is going to cause hyperpolarization. So after all of that, we have our photoreceptor right here. We have the cGMP gated channels closed because of everything that happened over there. So cation influx stops, and this photoreceptor hyperpolarizes. So again, this photoreceptor, everything that you just saw occurring over on this side occurred all within this cell right here. And step one is the cGMP gated channels are going to be closed because of the cGMP is off. So the cation influx stops and the photoreceptor hyperpolarizes. Step two, so this is when you can go and you can think back about how the NMJ works. And that's going to help you kind of understand this conceptually. Because step two is when those voltage-gated calcium channels on the synaptic terminals of the photoreceptor are going to close. Okay? Remember, in the NMJ, you have your voltage-gated calcium channels on the axon terminals of the presynaptic neuron. And then if an AP comes down, it opens them up, causing calcium influx to interact with the vesicles to release the neurotransmitter. Well, none of that's happening here because you have a hyperpolarized cell. Therefore, no neurotransmitter is released to bind to the next cell, which is the bipolar cell. So, the lack of iPSPs in the bipolar cell is going to result in depolarization of the bipolar cell. So, once you get to the bipolar cell's axon terminals, you now have an action potential because you have the depolarization. That action potential opens up those voltage-gated calcium channels on the synaptic terminals, allowing for calcium influx to come in, which then allows for EPSPs to be released onto the ganglion cell, which will cause an action potentials, many of them, to be fired along the optic nerve, cranial nerve 2. And so this picture is just an extension of the optic nerve right here. So imagine this is going all the way up. You have your eye, optic nerve. The action potentials are going down. It's going to cross the optic chiasma, which is right about here. Going to be filtered to the thalamus right here. And then it's sent to the primary visual cortex in the occipital lobe. And remember, it crosses. So If that light is picked up in the left eye, it's going to cross to the right primary visual cortex in the occipital lobe, and vice versa. Remember, the occipital lobe is in the back of the brain. All right, so this is how the dark pathway works, okay? So essentially, with the dark, you're not gonna have photons hitting visual pigment, so none of these steps are going to happen. This ain't gonna happen, this is not gonna happen, this won't happen, okay? This won't happen. The CGMP is going to stay on the CGMP gated channels because the PDE is not activated. And so, that's going to keep the channels open, allowing for sodium and calcium to influx the photoreceptor, which depolarizes it. And so since the photoreceptor is now depolarized, we have the voltage-gated calcium channels open in the synaptic terminals, allowing for that calcium to come in. Neurotransmitters released continuously, and this neurotransmitter is going to cause IPSPs to be released in the bipolar cell, hyperpolarizing the bipolar cell, closing voltage-gated calcium channels on the bipolar cell's axon terminals, which inhibits neurotransmitter release. Therefore, no EPSPs will be released and occur on the ganglion cell, and so the ganglion cell does not fire any action potentials along the optic nerve. So nothing travels through here, nothing travels through here, nothing hits the cortex, and you just see dark blackness.